Welcome to this complete masterclass on phylogenetic trees, diving into evolutionary biology, taxonomy, and the visual language of descent. A phylogeny is a branching diagram representing the evolutionary history of organisms. It shows how different species descended from common ancestors, demonstrating a branching pattern of divergence over time, rather than a ladder of progress. The basic architecture includes tips or leaves, which are the organisms being compared. Nodes are the branching points, representing speciation events. Branches are the lines connecting nodes, showing genetic lineages over time. Topology is the specific branching pattern. Branches can be rotated at nodes without changing the relationships. Remember, reading across the tips left to right doesn't indicate relatedness. Only the branching order matters. To trace ancestry, you find the most recent common ancestor, or MRCA. This is the intersection point when tracing two species branches back. Species with a more recent MRCA are more closely related. We now move to part 2, where we discuss taxa, clades, and shared characters. A taxon is any named group of organisms, existing at any taxonomic rank. In a phylogeny, taxa are usually at the tips of the branches for comparison. A clade is a valid evolutionary group that must be monophyletic. This means it includes a single common ancestor and all of its descendants. You can identify it by making a single cut on the tree. Invalid groupings include paraphyletic groups, which leave out some descendants, and polyphyletic groups, which do not include the most recent common ancestor. Organisms are grouped into clades based on synapomorphies. These are shared, derived characters that evolved in the ancestor of the group, such as hair and mammary glands defining the mammalia clade. Next, we delve into part 3, rooted versus unrooted trees. Rooted trees have a single basal node, the root, defining the direction of evolutionary time. This establishes ancestral versus derived states for the included taxa. The root represents the oldest point. In a universal tree, this is the last universal common ancestor, or LUCA. All life traces its lineage back to this single node. Unrooted trees display relatedness without indicating a common ancestor or a time axis. They do not show which nodes are older or younger, and are often displayed as radial networks. Tree-building algorithms typically create unrooted trees first, based only on data similarities. Rooting requires extra biological assumptions or external data, and a single unrooted tree can lead to multiple rooted topologies. We now begin part 4, covering the differences between scaled and unscaled trees. A cladogram is an unscaled phylogenetic tree. In cladograms, branch lengths are arbitrary and convey no numerical meaning. They only convey the branching order, or topology, and clade groupings. Phylograms are scaled trees, where branch lengths are proportional to the amount of evolutionary change. A longer branch indicates more significant genetic or morphological shifts, providing a visual gauge of evolutionary rates. Chronograms are time-scaled trees, also known as ultrametric trees. Branches are scaled to represent absolute time, such as millions of years, and tips representing living species align at the present day. Chronograms often use the molecular clock hypothesis, assuming DNA mutates at a constant rate. By combining this rate with fossil evidence, scientists can precisely date the divergence nodes. Moving on to part 5, we will discuss operational and hypothetical taxonomic units, known as OTUs and HTUs. Operational taxonomic units, or OTUs, are the observable entities at the tips of the tree. They can be species, populations, or DNA sequences, and are the raw empirical units being compared. The placement of OTUs is determined by analyzing observable data matrices, such as aligned DNA or protein sequences. Differences in these sequences drive the computational algorithms that build the tree. Hypothetical taxonomic units, or HTUs, are the internal nodes. They represent inferred ancestral states that are no longer extant and are statistically reconstructed based on the descendant OTUs. OTUs are real, measured, and empirical data, while HTUs are calculated, theoretical, and ancestral. Phylogenetics uses the known OTUs to accurately predict the unknown HTUs. Finally, we arrive at part 6, focusing on sister groups and the outgroup. Sister groups are pairs of taxa, or clades, that are each other's closest relatives. They diverge from the same immediate common ancestor and are considered the same age. Identifying sister groups is crucial for comparative biology. For instance, humans and chimpanzees are sister taxa because they share a more recent ancestor with each other than with other primates. An outgroup is a closely related taxon that branched off earlier than the group of interest, the in-group. It serves as a reference to determine ancestral versus newly derived traits.